Okay, today we'll be talking about the immune system and non-specific immunity. There are two types of immunity, and the next section of notes will actually focus on the specific immunity. Okay, let's review some terms from biology. First of all is a pathogen. A pathogen is any microscopic organism that can cause diseases. This can be viruses, bacteria, a fungus, parasites, such as worms and such. Some other things like, for example, prions, which are these proteins that actually rearrange the proteins of the body and cause them to become infectious. As a matter of fact, prion stands for infectious proteins. Sometimes they can be molecules, such as chemicals, some types of metals in the environment, or even like allergens like pollen or maybe even peanut dust. And each of these things attack in a specific way. In other words, they have a specific way of annoying your body and causing an allergic response. Okay, so the immune system is divided into two major categories: innate immunity and acquired immunity. And innate immunity is very similar. Basically, it is the same term as non-specific immunity. The immune system then is a network of lymphatic organs, many tissues, and cells and cell products. The purpose of the immune system is to keep the body healthy, keep it in a homeostatic balance. So that's what a disease is. A disease is a state of homeostatic imbalance due to infection or immune system failure. So you have your non-specific, that's innate. That is just keeping things out to begin with, and specific immunity or specific defenses, it's acquired. This is an example of your body learning how to fight something off, learning, hey, I've been exposed to this, here's how we're going to take a specific plan of attack. Okay, the first of the non-specific defenses is going to be barriers to entry, and this is going to keep things out to begin with. We have the inflammatory reaction. That's the next one. Inflammatory reaction is pain and swelling. You have phagocytes, which are large white blood cells that are going to eat any of the pathogens. We have the natural killer cells, which are pretty nifty themselves. They're going to poke a bunch of holes in any cell that's not functioning properly. Protective proteins, basically proteins that help with the immune response. And the last thing is fever. We can even raise the temperature of our body to try to bake the little pathogens out of us. Okay, barriers to entry. Skin is the first one. It's going to keep the bacteria on the outside and keep the good stuff, us, on the inside. You have ciliated cells, which are going to be cells with tiny hair-like projections that just catch bacteria and pathogens as they come into the body. Mucous membranes, basically snot, boogers, earwax, any orifice you have has some type of mucus that's going to be, the sole purpose is to trap bacteria and viruses and other pathogens from getting into you. So and that's what coughing is. Coughing is just expulsion of that mucus, and digestion. You have stomach acid, which will actually destroy pathogens. You have secretions on the skin, such as sebum and oil, and again, that's to just trap the bacteria and viruses. Sometimes these our pores get clogged and the sebum and oil build up underneath the skin, and that's where we get pimples. And then also competition with beneficial microbes. We have microbes in our intestines and on our body that are beneficial to us. They do things like help us produce vitamin K or absorb vitamin K. They help us digest, break down starches and complex compounds. And these beneficial bacteria actually keep down the numbers of the bad bacteria because they're in constant competition for the resource being you. Okay, so more secretions, chemical barriers. We've already talked in depth about mucus. We also produce lysosomes, which are little packets of digestive enzymes. So your saliva contains digestive enzymes, and these can actually even start digesting things like bacteria. Lacrimal fluid, even breast milk has lysosomes in it. Antibiotic properties there. High acidity, like for example our stomach, gastric juices, a pH of only 2, very, very strong acid, urine, vaginal secretions. When we talk about reproduction, we will talk about how the sperm cells actually have to make adaptations to be able to try to survive in a vagina, because it is hostile to even sperm cells, but particularly like bacteria and such. And then sebum, again, that oily substance that's produced from our skin to help keep bacteria out of our pores. And then if they get clogged up, we have problems. Okay, so if you look at this clip here, you can see there's something definitely wrong with the right hand compared to the left hand, and this is the inflammation reaction. Something has happened there. It could have been a bee sting, it could have been they were swinging a hammer and missed the nail and hit their hand instead, but something has gone wrong there, and so damaged cells release histamine, and histamine causes swelling, heat, redness, pain. And all of this is to help the immune response. Basically, the capillary is enlarged, so if we look down here, we have a smaller capillary. There's the damage from a pin, and so the cells here that are damaged release histamine, bacteria are in there, the capillary gets larger, and that causes more tissue fluid to get to the area to help immobilize, as well as allows these blood cells to then pass through the capillaries more easily, this process known as diapedesis. So migration of the phagocytes, and it says right here, amoeboid in shape to the tissue. So in other words, these macrophages or big eats get in there, and then they start gobbling up the bacteria. Okay, so cellular defense, here we're going to talk about phagocytes, the macrophages, or the big eat, and these things actually gobble up other cells. We can see it right here. It's wrapping around the bacteria and digesting it. Neutrophils, eosinophils, and basophils all are different types of phagocytes that will eat other cells. The basophils particularly are the mast cells that we see in other tissues, such as like the nervous system or the skin. And then the last one here we have are the natural killer cells, which actually will bind with cells that are infected or have other issues like cancer, and they release perforins and cause them to burst. Okay, so here we have the phagocytic mechanism, basically what happens now after we have this puncture here, the bacteria is in, the histamine acts like an alarm call, and that causes the vasodilation or the enlargement of the blood vessels. Margination basically means that these cells become more spread out and there's more of a gap there. Diapedesis is the movement of the cells across into the cell fluid, the cell tissue, and chemotaxis means movement towards the chemical, particularly this chemical is histamine. Histamine actually draws these bacteria in. Another side note I have forgotten to mention is that histamine is something that causes swelling and pain. It's the same chemical that's released when you have a stuffy nose, which is why your nose becomes all swollen and hard to breathe. And so if you take an antihistamine, it reduces that swelling. Antihistamines also reduce things like bee stings and other things where you see pain. Sometimes it can do things like help with like even a twisted ankle and such. You take a pain reliever that has an antihistamine property to it. Here we have the phagocytic response, which basically just means that this phagocyte or large eating white blood cell is digesting something. So the pseudopodia are found here and here, actually here and here as well, and so they're wrapping around these bacteria, and then it pulls it in and it ingests it in this vacuole, basically a little bubble of cell membrane that was brought in, and it fuses with this lysosome, which is a little packet that contains digestive enzymes, digests the bacteria. Oh, I'm melting, and now it's dead, and now exocytosis, it goes out, much like defecation, but instead just this little bubble fuses with the wall and is released. So here are the natural killer cells, one of the coolest named cells in the human body. They kill virus-infected and tumor cells by binding to them and releasing perforins, which poke a bunch of holes in it. They are granular lymphocytes. These guys are not specific. They're just looking for anything that's causing problems in the body, anything that's not normal. There's no memory here. Again, it's not specific. It's just looking for something that's causing a problem. And again, they release perforins, which is poke a bunch of holes in the cell that's malfunctioning, that's infected, and causes it to basically burst. Okay, the fourth one is the protective proteins, and here we have the complement system. These are blood plasma proteins that activate an immune response. I'll go over a little bit more with the complement system here on the next few pages. But they're going to do things like amplify the inflammatory response, aid antibodies in what they do by causing things to clump together or poking holes, form attack complexes, which again is poking holes. And then you also have interferons, which are these chemicals that are released, and they basically tell non-infected cells, hey, prepare for attack, don't bind with the virus, it's just trying to trick you and do harm to you. These are also species specific, meaning specific to the type of virus. So I'm not overly worried about you having a firm grasp on this. I want you to sort of have an appreciation for the complexity and sort of have an idea of what some things that happen. So this is a complement system, an example of one. So here you have chemical C3, which when it binds with C5, C3 splits into C3b and C3a. C5 then splits into C5b and C5a when it binds with C6. And then C6 combines with C7, C8, and C9. Now C6, 7, 8, and 9 cause holes to be poked into the cells of whatever it's attacking. That causes fluid to go rushing out, and the cell dies. C3b actually will leave C5 and attach to these bacterial cells, causing them to be marked for death, more or less. It's kind of like an alarm. It attracts the phagocytes here, and here is these pseudopods, and it's going to wrap around and digest those bacteria. Makes them a little easier to identify. The other thing is C3a and C5a will then go on to increase inflammation, which basically draws in more of the white blood cells, more of the macrophages to eat up the bacteria or whatever's causing the problem. So more or less you have this cascade of events, and one thing they do is mark the bacteria, they poke holes in the bacteria, and they draw in more white blood cells. So today we've talked about innate immunity. Next section of notes we'll talk about acquired immunity in depth and in detail, but we will start to mention some of the stuff today. Particularly we'll talk about some of the humoral response or antibody response. Okay, there are two types of specific immunity. We have antibody-mediated, which is going to involve B cells, and cell-mediated, which involves only T cells. Both involve helper T cells. An antigen is a surface molecule. It stands for antibody-generating protein, and so it's anything that causes a reaction. So we have self-antigens, which are proteins on the surface of our cells. They can be found on organs, or like in the case of blood cells, they're the A, the B proteins, the pluses, the Rh proteins. So for example, in a person who's AB positive has the A, the B, and the Rh proteins, whereas O negative has none of those. We have non-self-antigens. Those are things that are found on other things like on bacteria, viruses, or it could even be something like peanut dust or pollen or something like that that's going to stimulate an immune response. Again, antibody generating is antigen. We have B lymphocytes. They are produced in the bone marrow and mature in the bone marrow. They produce antibodies when they become plasma cells. T lymphocytes directly attack cells. They become what are known as cytotoxic T cells. There's also the helper T cells. T cells mature in the thymus. They start in the bone marrow, go to the thymus, and mature there. So all cells have some type of surface macromolecular protein to identify them. Adaptive immunity is sensitive to these specific infections based on antibodies fitting these specific macromolecules or proteins usually. It uses a lock and key method. In other words, see how this thing is sort of has a sticky, pointy out bit that's triangular shaped, and the antibody has a triangular, pointy in bit that's triangular shaped. So that's what the lock and key method refers to, and it basically just means the antibody will fit perfectly with that macromolecule and attach it to identify it. Antibody-mediated immunity, also known as humoral immunity, uses the B lymphocytes. So we have what are known as antigen-presenting cells, or APCs. They're going to present the antigens from the pathogen to a helper T cell. The helper T cell then stimulates these B lymphocytes, which are made in the bone marrow, and they recognize the antigens directly. Basically, when a memory B cell bumps into the bad guy, again, let's call it the pathogen, then it will start cloning itself and then start releasing antibodies immediately, which allows us to basically have cellular immunity that's memory. It's memory immunity. So here's your basic structure of an antibody. You have what's known as a constant chain. This always stays the same. And then you have the variable chains here and here. It means that varies. So every antibody has this constant chain or heavy chain. It's really, really big. The light chain or variable chain found here and here changes based on what it's attacking. What does it fit with? The shape changes here to like a triangle if it's a triangular shaped protein, or more circular if it's a circular shaped protein, and so on and so forth. All right, so the major classes of antibodies. The first, there are immunoglobulins, Igs, and the first ones are IgG. IgG are monomers, meaning there's one of them like this at a time. They're Y-shaped, and they're the most common. 75 to 85% of antibodies are IgGs. They bind to the pathogens and toxins or things in the body that cause problems. These are the only ones that cross placentas. So in the erythroblastosis, or basically where the mother is allergic to the Rh compound of the infant, these are the ones that are coming into play there. These are ones that provide immunity for the infant while it's in utero. All right, so here we have IgMs, and these are pentamers, meaning there's 5 of them. So here's your pentamer. These are activators of the complement system, so they get that whole protein cascade going. These are released early in infections. These are a primary response. They're a agglutinating agent, so when you have the blood transfusion reaction, they cause the blood cells to clump together. But again, these guys are released in infections. And the primary response in agar, and also clump like bacteria and such together, making them easier for a white blood cell to digest one of those macrophages, because it's easier to get a whole group of them at one time rather than get each individual cell. IgAs can be monomers or dimers, as you see here. These are found in body secretions like in saliva, sweat, tears, and digestive juices, and breast milk. One of the first things that doctors would always ask when we brought in our infant children into them when they were sick was, are they breastfed or formula fed? And formula fed, the children are not going to get the antibodies, but breast milk does provide antibodies from the mother and gives a little boost to help immunity for the children. As a matter of fact, one of the parenting books we read before we had children told us that you could squirt breast milk up a baby's stuffy nose. A little odd, but it is something that has been found to be effective, because the antibodies will attack anything that's causing problems in the nose. IgDs are always monomer, so here's our IgDs. And they are found on the surface of B cells, and they're used in the activation of the B cells. IgEs. So here's an IgE. IgEs are always monomers. They're secreted by the mucus of the GI or digestive tract and the respiratory tracts. So anywhere we see mucus, that's where we're going to see IgEs. They stimulate the release of histamine, so if something gets in your stomach that you don't agree with, or something gets in your lungs, like for example pollen, and you're allergic to pollen, they can stimulate the release of histamine and cause an allergic reaction, so respond to allergies and parasites. And to remember the different classes of antibodies, just remember this word here, MAGE. You have IgMs, IgAs, IgDs, IgGs, and IgEs. So here are some examples of how these antibodies can work. So for example, an IgA can bind and neutralize these toxins or chemicals that are in the body, and then they can be ingested by the macrophage here. IgGs, they can help bind and block this bacteria from doing what it's doing, and so the macrophage then can more easily gobble it up and locate it and digest it. IgMs sort of kind of do the same thing, but can actually cause a whole bunch of them to clump together, and also set off the complement system, which causes these things to burst. If you remember, we were talking about those at the end of that complement system a few slides back. They would also burrow holes in it as well. So another look at some different ways that antibodies work. Neutralizing it, basically wrapping around it. See, this virus now can't enter a cell because it's been wrapped around. Agglutinating, causing things to clump together, makes it easier for white blood cells to digest. Precipitation, here's a chemical that when it's dissolved can enter the cells and cause problems, but when it's precipitated, it's been neutralized. Activating the complement system, which can do things like poke holes in the cells. They will enhance phagocytosis, making things easier to find. They can enhance inflammation, again, increasing the immune response, and cell lysis, causing the cells to actually burst. All right, so the next section we'll talk about the difference between humoral and cell-mediated responses. But basically what's going to happen is you're exposed to whatever's causing the disease, the antigen, the pathogen. Then it's going to be displayed by dendritic cells, also known as APZ, or antigen-presenting cells. They activate the helper T cells, and if it stimulates a B cell, it will divide and produce some memory cells, but also produce plasma cells, which then release antibodies. The helper T can also stimulate cytotoxic T cells, which bind directly to infected cells and release perforins and poke a bunch of holes in it. They also release memory cells too, so that way if any of this stuff happens to show up again, we have these memory cells, and they basically eliminate this whole process. They just immediately start dividing and start releasing antibodies or start attacking the cells directly. Okay, I would like for you to post a comment to show that you have actually read these notes and viewed this presentation. So somewhere in the comments, just go ahead and post something. It doesn't have to be anything big, but please post something to show that you have indeed read this for attendance purposes. Thank you.